Bismillah Rahman Rahim. Welcome again, guys. Welcome to another video's lecture from Biology. We talked about the replication schemes of different viruses. We talked about the replication schemes of the DNA viruses, RNA viruses. Okay. We also talked about the pathogenesis of the viruses. If you remember, we talked about the naked viruses, means how the naked viruses enter inside the host cell and how the envelope virus enter inside the host cell, but we didn't talk about their release processes, that how the naked viruses are released from the host cell and what happened once they released, and how the envelope viruses are released from the host cell and what happened inside the host cell when they released. Fine. So in this lecture we will talk about the release process, means the release of the naked viruses as well as the release of the envelope viruses. When they release, then after that what happened inside the host cell and what happened to the host cell. Host cell die or host cell not die live. So what happened? So we'll be talking about all in this video lecture. So now let me write, we will talking about the release of naked and envelope viruses from host cell. So how the naked virus and envelope virus are released from the host cell. Let me draw a line between them by which we can easily understand the processes between both of each. Here we will discuss the naked viruses and here we will discuss the envelope viruses, that how the naked viruses and how the envelope viruses are released from the host cell. As you better know that naked viruses are those viruses which don't have the envelope. They have just capsid around the nucleic acid. They have just capsid around the nucleic acid and they have the nucleic acid, DNA, RNA or any kind of the genetic material they have. Okay, so they have two things. But envelope virus, as I told to you, they have three important things. One thing is envelope, outside membrane area, and the other thing is the capsid area, capsid, and within the capsid they have the genetic material, RNA or DNA or any kind of the genetic material they have. Now let's talk about that what happened. I already talked the pathogenesis, means the attachment and the engulf. If you missed that video, so go back to my channel and watch that video, and after that then you will see the release of the naked and envelope viruses from the host cell. Now what happened? This is a virus. Okay, let's suppose this is a virus. They have just, this area is called capsid, and inside this capsid they have the genetic material. This green one represents, let's suppose, the genetic material, RNA, DNA, any kind of a genetic material they have. Okay, now what happened? Let's suppose this blue one area represents the host membrane. They attach to the host receptor. Once they attach to the host receptor, so they will be engulfed. So I already discussed, but let me again just summarize to this process. So they will be engulfed. So we have structure just like that. Here we have a naked virus because they don't have envelope, and having the genetic material as well. After that what we get a complete engulfation of the naked virus. So finally we see that the naked viruses, so we see that endosomes are made. around the naked virus. So we get an endosomes. After that, they just release their genetic material, means DNA, RNA, any kind of the genetic material, and with this genetic material they also produced their structural protein. Let's suppose this black one represents the structural proteins. Now with this structural protein, what we get? Finally we get a progeny. By the combination of this genetic material and this structural protein, what we get? We get again a progeny virus. Fine. So we get again a progeny virus. This is a simple pathogenesis, means attachment, engulfation, and their release and their replication, all this. And after that, what happened? If I draw the host cell, so what happened? Inside the host cell, there will be also the genetic material of the host cell as well. They have the genome inside the host, their own genome as well. And there will be a naked viruses. So a lot of the naked viruses, means progeny viruses, are made, and once they made, so what happened? So after that they will just, there will be a genetic material, DNA, RNA, or any kind of the genetic material they have. Now what happened? They will put pressure, they release some enzyme, lysozyme enzyme, and once they release that enzyme, so the cell will be degrade, the cell membrane will be burst, and when the cell membrane burst and the cell will start to degrading, so all the viruses will be released. So here the most important thing is that the naked viruses will kill the cell, will lyse the cell, will die the cell, and then these naked viruses are released. So they are released during cell lysis. Cell lysis. After that, the cell will be lysed, cell will be die, cell will be killed, and cell will no more long function because cell is already die, and the all viruses are released and ready to attack another new host cell and repeat the whole process once again. So that's all about the naked virus. Let me write. That's all about the naked virus. So that's all about the naked virus. Now what happened in case of envelope virus? In case of envelope virus, they will be enter inside the host cell through a membrane fusion, as I told to you. So if I draw the naked virus, so again we have a virus. They have the genetic material, but they have the envelope as well. Yes, this black one represents the envelope area. So after that, what happened? This is a membrane of host membrane, and they will be attached to the receptor. They will attach to the receptor, and after that, what we get just like this form. Sorry. They will be fused with the membrane. So what happened? They will just fused with the membrane. Means the envelope area of this virus will be remain outside, while the capsid area of this virus will get enter inside the host cell with the genetic material, having the genetic material. So they will be enter with the genetic material inside the host cell. So finally we get the structure just like that. The membrane structure will be just like in this form. So the virus, our virus is now present here. Again, this is a host cell. With this host cell, what happened? Let's suppose this is a host cell. Now what happened after that? This envelope virus once made their copies, means once the progeny envelope viruses are made, then what happened? They released some sort of the protein, some kind of proteins they released. Let me draw the proteins with this black one. So they release some kind of proteins, and these proteins will be attached to the host membrane. Now once these proteins attach to the host membrane, so this progeny virus will attach their self with this. So they will attach their self. So what we see just like this form diagram. Once again, let me draw. So we see that our progeny virus are now attached with this protein area. Now they attach with this protein area. Once they attach with this protein area, then what happened? After that what happened? So they will be make up. If I draw the structure of the cell membrane, so there will be a structure just like in this form. And now here is our virus. Having the genetic material, DNA, RNA, or any kind of the genetic material. And here is the protein as well. Now they attach with the protein, okay? So they attach with the protein, and finally we get the virus. So there will be a punching. What does mean punching? Punching means there will be a punching occur, means this area and this area will be closing to each other. So we get just like that structure. So we get structure just like that. There will be a protein. So punching may be occur, means the cell membrane of the host will close this area and this area. I mean this and this area will close to each other. This is called punching of the cell membrane. So once they punch, so our virus will get enveloped. So finally we get the structure of the virus just like that. Finally we get the structure just like that. Means how? Let me draw once again. Or we can also say, so our virus will be look like in this form. They have the envelope. There will be our virus structure, envelope virus. So we get an envelope virus. And having the genetic, they carry the genetic material, and these are the protein receptors. They have the protein receptors. So these protein receptors are now attached with this and finally we get an envelope viruses, this or this. Once again, explain me the whole processes for your understanding. This envelope virus will not kill the cell. They just kill the cell at the end, but not from the beginning. The envelope viruses are released through budding. So what happened? Budding, released through budding, released through budding. So they are released through budding. Fine. What happened from the beginning? They are released through budding. Means this is a budding process. So what happened from the beginning? Let me again explain the whole process for your understanding. This is a naked virus. They have just capsid and the genetic material. They will attach to the host receptor and engulfed. And once they engulf, so endosomes will be made. Endosomes will finished and just the genetic material will be released, and this genetic material is further used to produce a structural protein, viral structure protein, and then we get a progeny viruses. Now this is a host cell, and this progeny viruses will put pressure and release the lysozyme enzyme by which the cell will be killed. So they kill the cell, and after killing the cell, these viruses will be released. And what happened in this case? In this case, let's suppose we have the envelope virus, and the envelope virus will be entered inside the host cell through a membrane fusion. As I already discussed this, so go back and watch that video. So this will be fused with the membrane of the host cell. When they will fuse, so the envelope area will be remain outside while the virus will get enter, and they just release their genetic material. And after the genetic material, they also release some protein, and this protein will linked to the host cell membrane. Once they link with the host cell membrane and we get the virus particles, these virus particle will attach themselves to the host, means the protein with their protein, with the virus protein. So when they attach with their own proteins, so what happen means the punching process will be happen. The membrane area will be close to each other, just like that. This area and this area will be closing to each other. We call that punching, and after that we get enveloped. We get the envelope, and this containing the protein of the viral is viral's own protein. So we get envelope viruses, or we get this just like that structure, or just like that structure. So finally we get envelope viruses. So does the difference is just like there is a very short difference. Means they will enter in this process, mean endosomes we get, and they will enter just a membrane fusion, means the envelope area will be outside. And other difference is that when they release, so they will kill the cell, lyse the cell. And when the envelope viruses are released, they just release through budding. They not kill the cell, they not harm the cell. Means they not degrade the cell. They are just released through the budding processes. And the rest of the viruses also released through this budding process. Okay, I hope you understand all of the idea. If you have any kind of question about this, so write a question in a comment. I will try my best to give you answer as soon as possible. Okay, so I hope this video helpful. If you like the video, please set the like button, share this video, and there will be a subscribe button, and please subscribe the channel to get more interesting video lecture like that. Thank you so much.